Hey guys, welcome back to the show. Today's episode is, it's super exciting. This is one of those ones where it's a problem that we've all been looking for a solution for a long time, and it is finally here. We're going to be talking about a relatively non-invasive, accurate test to test mitochondrial function. And there's been very few options in the marketplace. And finally, we have something that does not involve using a tool to punch a hole in your muscle, which is as unpleasant as it sounds. Okay, but before we jump into the episode, number one, gratitude for you for being here. Thank you for checking in. Thank you for being here and listening to the podcast. We are growing like crazy this year. I know we have a lot of new listeners, so I want to welcome the new people. I want to thank people who've been avid listeners for a long time. I appreciate every last one of you. If you are getting value from these episodes, please make sure that you share them out with your friends, networks, family, anybody who you feel would also get value from them. And please make sure that you take a moment, when you have a moment, to leave us a review on whatever platform you are listening to this podcast on. And everything else that I normally talk about, the newsletter, the membership community, all those things, you can go to natniddam.com, and that is where you will find details on all of these things. All right, let's talk about this incredible episode. We know that measuring mitochondrial health has always seemed really tricky. I mean, it's been a problem, right? We know that people go into labs and they do these crazy experiments. They punch a hole in people's muscles and then they extract muscle tissue and they look at the mitochondria. Number one, that's expensive. Number two, it's really painful. And who wants to do that? Now, not anymore. You don't have to do that anymore. In this longevity episode, I am joined by Dr. Hemal Patel to discuss groundbreaking research that allows us to assess mitochondrial function through a simple at-home blood test called the Mescreen test. We discuss, and then we go in to discuss all of the many crazy things that you would never imagine actually have positive impacts on your mitochondria. Things like mind practices, things like immersive retreats, and how Dr. Patel and his team have actually quantified this stuff. So we also dive into talking about the cell and Dr. Patel's personal story of how he integrates meditation into his nightly routine for better sleep and creativity. So Dr. Patel has a beautiful resume and bio that I suggest you look up in the show notes, because I don't want to stand between you and your podcast episode any further. But I do want to tell you that if you decide that you want to get this test, we can save you some money. All you have to do is go to mescreen.test and use code longevity. And now let's jump into the episode. Hey folks, just a quick reminder that all of the information presented in this podcast is for information purposes only. No medical advice, no diagnosing, no treatments suggested here. Before you try anything that you hear about or learn about here, make sure that you check with your medical provider. Welcome to the show, Hemal Patel. This is going to be exciting.
Good to see you.
I'm stoked to have you here. This is one of those conversations. I'm like, I've always really wanted to do an episode on mitochondria and measuring it and this and that, but I could never figure out who to do it with. And then I met you. I was introduced to you by a mutual acquaintance somewhere. I was like, yes, finally. And I think it's been a long time coming, right?
It has. I mean, and we've been doing this in the lab for over a decade, and who knew humans would actually want their mitochondria tested, right? We've been doing this in mice and rats forever. Turns out humans actually want to know as well, which is great.
Well, they do now. I think maybe 20 years ago they didn't so much, or maybe 50 years ago they had no clue why they might even. So, which actually brings up a really good first question is why? Why would we want our mitochondria? Why would we want to know what our mitochondria are doing?
Yeah, I mean, it's a huge buzzword right now in biohacking, in longevity, in all of these places where people want to get healthy. They realize that the mitochondria are a core element of that health. And so you think about where they are, right? They're in every cell of your body except the red blood cell. And so they regulate every organ. And they are sort of pedantically referred to as the powerhouse of the cell, which is true. They make a lot of energy and they make it in an efficient kind of way. but they do way more than that, right? They're the gatekeepers for cell death and survival. And so this is where they become really important in either your limited life or your endurance resilient life as well. And so if you make your mitochondria better, you can get not only the energy profile, but your cells are living in a much more healthy state. And this is where I think the real turn on this is having key mitochondria that then regulate this. One of the things my lab has been interested in studying for many years, I've been at UCSD 22 years now, is non-communicable chronic diseases. So if you look at every non-communicable chronic disease, the end effect of all of that is an energy demise, right? You're not making enough energy to supply the demand that the body or that organ has to keep up. And it's by design, right? There is no body battery. There's no way to store ATP. And so as soon as you make it, you have to essentially use it to do the functions of life, and then you got to make more of it. And so your mitochondria never stop. They're always active, and organs like the heart makes 2 to 4 kilograms of ATP a day, yet you're not increasing your weight that much, right? You're constantly churning this around. And so a system that has to work from the minute you're conceived to the minute you die has to be working at an optimal level. And this is where you get sort of a regulatory and demise of systems. Is every endpoint of these chronic diseases leads to a mitochondrial dysfunction that then leads to end organ failure and organism failure. If you can somehow minimize that mitochondrial dysfunction, you tend to have a very different effect in that chronic disease. And this is where I think we're trying to learn and intervene. And this is what most humans are sort of dealing with.
Yeah. So you're saying the end point, but is there ever a case where the starting point, is it sometimes the first place that starts to go down, which, you know what I mean? Like you're talking about the mitochondrial dysfunction being the end point, but is there ever a case, I think people do believe that sometimes it starts with mitochondrial dysfunction, which leads to disease, which makes sense, right? Because you start to run out of steam. The cells can't perform the functions that they need to perform.
Right. There's some evidence for that, right? And we've been looking at it from a unique perspective. So if you think about your mitochondria, back about 1.5 billion years ago, they were old bacteria that got incorporated into cells. They gave a cell a unique advantage over other cells. It was now a symbiotic relationship where you're sharing a space, and it's a contract that we made with them back long ago. They provide this huge burst of energy, which then allows us to move, grow, become complicated, and do crazy things. That evolution, I think, is an important aspect of this. This is this endosymbiotic theory where chloroplasts and mitochondria came from this sort of rudimentary source. One of the things that we've been thinking about how that dynamic shifts is there's a huge focus on the gut microbiome these days, right? The gut is a control point for everything. And people call it the secondary brain, but in lots of research we've done suggests it may be the primary brain. It's regulating everything else. And so the big question is, how is your gut communicating with your body? And if you think about what likes to communicate with each other, like likes to communicate with like. So your gut bacteria, the things in your body that they can relate to are your mitochondria, because they're ancient relatives, right? And so we think that that's the communication highway that's happening. And this is where diseases may start in this gut microbiome space. And it's this sort of relationship with the energetic profiles you have in your organs that may be at the primary end with this, right? So a lot of diseases you could link to gut as well. And so I think this is where that dichotomy comes. As we've been studying this in my lab, we have a sort of a different spin on the biology of the cell and what we think is primary versus secondary. So I trained as a cardiac physiologist. I then came to UCSD and did really high-end cell biology because I wanted to have the depth in my lab to be able to go all the way from, human, animal to molecular studies. And so as we started studying the cell, it turns out that the membrane, and we did this really silly survey on campus. We took a picture of the cell and went and asked people, what's the most important part of the cell? And as we're taught to think about this, right? The center, the control center, 64% said the nucleus is the most important. This is how we are taught to think about the world, that you have to go to the command aspect of it, right? Only 20% said mitochondria. And out of all of that, only 7% said the plasma membrane is the most important part of the cell. If you think about the cell, you can't have a cell, you can't have life unless you have a plasma membrane.
Yeah.
Because it has to limit a space and it has to limit the outside from the inside. And so my thought on chronic disease is it's when the outside now starts to intervene on the inside, right? And the primary cause of that is the destruction of the plasma membrane in one way or another, either through lipid reorganization, age. It could be mitochondrial-driven lipid peroxidation, which then, you get free radicals that then start attacking lipids in your membrane. So there could be a communication highway that way. Mm-hmm. But we really think that in lots of these diseases, the membrane starts to break down first, which then leads to this outside-inside disbalance. And then you start seeing this mitochondrial dysfunction that really sort of grows and ensues. And so a lot of the things we're doing therapeutically is fix your membranes. I mean, this is one of the first things we see in people who have really crappy mitochondria is forget about fixing the mitochondria, fix the lipids, right? You can fix the lipids in your plasma membrane, in your nuclear membrane, in your ER, and your mitochondria. That creates the foundation of health in a lot of these states, I think. So, lots of ways to think about it, right? The biology.
So that's fascinating. I mean, I had been taught in school that the membrane is the brain of the cell. So, which I think, it never left me in a way because the way they explained it was similar to you, but a bit different in the sense that that membrane decides what comes in, what goes out, and if what needs to get in can't get in, and the waste can't leave, and then waste comes in and good stuff comes out, in a very kind of basic way. Yeah. It's a non-starter, right?
It is. And it's the thing that responds to outside stimuli as well, right? It's the one that's sensing the environment that you're exposed to, whether it's positive, negative, toxins, all kinds of things.
Yeah. So it's really, it's interesting, right? Because you're almost saying, well, yeah, the mitochondria is our focus, but it's not, the mitochondria is not driving the entire agenda here. It's sometimes, or many times, maybe being driven by this, this kind of semi-permeable wall that defines the cell and allows it to control its environment, essentially.
And it's a true symbiosis. I think you need to have both things working in an optimal way. I mean, mitochondria likely get disgruntled as well, and you could potentially start from that endpoint. Yeah. And you see this in a very toxic way in people who have mitochondrial diseases, right? And so they have mutations in their mitochondrial DNA, which is, that's the other unique element of this is mitochondria have their own DNA, right? It's separate from your nuclear DNA. You typically inherit all of your mitochondria from your mother. And so you can actually trace back to the primordial Eve through mitochondrial DNA tracing if you wanted to. And so but that DNA is very limited, right? There's 37 genes that encode for 13 critical things the mitochondria need. When you start looking at the proteomics of the mitochondria, there's like almost 1,400 proteins in the mitochondria. So the bulk of the mitochondrial stuff comes from the nucleus, encoded in the genetics that comes from both your mom and dad. But then the core parts, the key things that the mitochondria needs to keep the functionality going come from the genetics that are trapped from your mother, right? From the egg that has the living mitochondria. And so people that have mitochondrial mutations in these particular sites have these morbid diseases, right? Their mitochondria don't work and they can't sustain life because their energy is compromised and their signaling is compromised. So there are ways that mitochondria could be primary aspects.
Yeah. So these are people who don't live very long, obviously.
Yeah. And the rate is fairly high. I think the numbers I was looking at is 1 in 5,000 people have some primary mitochondrial mutation in one way or another. Yeah. Not the severe kinds, but some level of it, right?
Yeah. So it's not fatal.
Right.
To that degree, but it's compromising.
Yeah.
And so how would that show up in these people? Is it metabolic dysfunction or
metabolic dysfunction, right? Just incompatibility with energetics and stressors and other things. And so people will express the mutations in unique kinds of ways. The horrific ones, there's a night and day difference, right? There's no way to miss them, right? They have impacts where you don't grow as well, impacts where you can't do all the functionality things that you need to do. And those are key aspects to metabolic fate that you can see. But the milder mutations, you can pretty much survive normally, but there are limiting things when you get put under stress.
And so for those, are those detectable in one of these commercial genomic tests, or do you need really?
So we've been looking at that.
Really fine tuned, yeah.
So if the SNP or the mutation is identifiable in the nuclear genome, you're likely to capture it. Mitochondrial DNA is unique in that it's low abundance, right? So when you isolate it, you're going to see less of it from that cell. There may be some contaminant participants that you could see. And we've been actually thinking about this is, can you create an array that looks at the genome versus the mitochondrial components to look at these aspects? Mostly people who have mitochondrial mutations, they'll go through specific genetic testing that'll look at those mitochondrial mutations. And there's tests to do that.
So they look specifically at mitochondrial DNA.
Yeah,
so
you can look at mitochondria or components in the nuclear DNA that could report into the mitochondria.
Huh. That's interesting. That's really interesting. And so that could sometimes explain why this person doesn't tolerate X or tolerate Y or...
And I mean, this is where you get this just heterogeneity in how humans express themselves, right? In their sort of physical and genetic space. Is you could have the most amazing mother who gives you the most amazing mitochondria with their genetics, but then you have a dad who gives you the short end of the stick in terms of the genetics, and your nucleus then informs the mitochondria, right? But then it could be the opposite, right? Your mom could have horrible mitochondria get passed down, but then your dad gives you these amazing 1,400 other proteins that populate. So it's a really complicated genetic and protein space, I think. And this is where the symbiosis becomes really complicated. Is now this organism, which was surviving on its own with this 37 genes, incorporates into a more complicated being, our cells, and it needs support from the nucleus, which has a much more denser genetic material to then do all these things that it needs to do. And so those 1,400 proteins now give the mitochondria functionality that they never had before. But at the same time, they have to live in concert with the rest of the cell, right?
Wow. I'm getting overwhelmed here. I don't know how my mitochondria feel, but I'm starting to get overwhelmed. And so, okay. So mitochondria have their own DNA that's influenced by the nuclear DNA, which is the much bigger piece of the puzzle, if you will. Yeah. Now, we often talk when, I've done a lot of, I've delved into genetic testing for people. And what I generally would tell my clients is, look, your DNA doesn't determine your destiny. Like it's maybe 10 or 20% how it expresses.
Right.
So the things that you do, the things you expose yourself to, and we're going to, at the end of this podcast, we're going to talk about a really interesting influence on DNA expression that people often don't think about. But it's those things that you do that really manage. Do you believe that that's also possibly true, aside from the catastrophic variants that really will take a person out?
Yeah.
Do we believe also that there's a piece of that as well? That a better.
I think there is, right? Your DNA is, and this is where the genetic testing becomes weird. It's looking at the potential that may exist. It's not the actuality. Yeah. Whereas measuring mitochondria is the actuality of how all of that comes together to manage functionality in your system. The other thing that becomes really complicated is if we start moving in this genetic scale, your mitochondria have 37 genes. Your genome, your nucleus, has maybe 25,000 genes. So we're very similar in terms of those aspects. You move to the gut microbiome, there's over 8 million genes that are being expressed there, right? So we're more diverse in our microbiome than we are in our genetics of our cells that we have. And so now you start throwing in this other entity that's living inside us that's expressing at a much higher level. And its influence on our biology is probably more dramatic than our genes' influence on our biology at the end of the day.
Yeah. It all begins and ends in the gut at the end of the day.
And they're going to be around after we're gone as well, right? So.
I know. You will live on in your gut microbiome. Yeah. Those buggers never die one way or another. Okay. So having said, so all this, we've taken people down this crazy rabbit hole. Let's pull back for a second and say, okay, so mitochondria, they were self-contained entities originally. Our cells made a deal with them. We evolved into humans one way or another. We're not, we're skipping a few steps.
Yep.
And in your lab, you felt that it was really important to try to get a handle on mitochondrial function, like how efficiently. And so, we talk about mitochondrial, the production of reactive oxygen species, mitochondrial respiration, all of these different things. So let's talk a little bit about what took you to this place of saying, look, this is the thing. We need to be able to measure this stuff. And there used to be this, well, there still is this thing where you can punch a hole in somebody's muscle, pull out a chunk of muscle, and actually physically get a sample. Highly unpleasant. Not practical. And probably
scalable, right? There's no way to scale something like that, right? So that's where we started. I mean, we've done hundreds of muscle biopsies on campus as part of clinical studies. It is the gold standard. If you're doing a research study and you're recruiting participants, it is the way to get access to direct mitochondrial functionality. We had a nice paper on Gulf War illness that came out that showed that it's really driven by a mitochondrial dysfunction that we see in these individuals over an inflammatory response, right? So it's not involving immune system function, but mostly mitochondria.
Sorry, what illness?
This is Gulf War illness. So these were veterans of the Persian Gulf conflict back in the 90s that were exposed to who knows what. And then they came home and had all these symptoms, brain fog, chronic fatigue, dermatological issues, those kinds of things. So it turns out the etiology of that is probably all of this exposure led to a demise of their mitochondrial systems, which then perpetuated all these years. So you get back into this idea of DNA versus your mitochondria. So whatever epigenetic trigger they had in the 90s, it's entrenched where we can still see it in their muscle, and we can still see it in cells we isolate from their skin fibroblasts, right? This phenotype that we're observing at the level of the mitochondria. So it's a powerful trigger sometimes, these environmental exposures that we have. So the muscle biopsies were critical. It's, you get direct access to someone's genetics, you get their mitochondria, and you're measuring their functionality. Scalability is impossible with it. We have a really well-equipped lab, and we could do maybe 2-3 a day. And you have to take a break because it takes 6 to 8 hours to analyze those samples. And the person running them is just exhausted because their head's cut off and they're doing all these crazy things to manage those 3 samples. So
and you've got people limping around. Let's be clear.
And so this came out of necessity. And so we were involved in the NASA twin study, where the twin went up into space for a year and his counterpart, the senator from Arizona, was on ground. And the task was, we can't give you any tissue. We can give you plasma. So invent a mitochondrial test that tells us when and in which organ system the mitochondria are going to be dysfunctional when this guy's up in space for a year. And so the brainchild I came up with was this idea that could the plasma be a representation of the individual in terms of their accumulated environmental capture, right? So our plasma, when you go into a doctor and they're, concerned about what you may have and they don't know what it is, one of the first things they'll do is blood tests, right? They'll look at your red blood cell count, white blood cell count, all these kinds of things. So blood has information that it captures across our bodies and from all our organs, all our cells. And so the thought was every biochemical reaction that's happening in our gut, in our organs, is being captured in some minutiae kind of way in your plasma, right? All the metabolites are being accumulated and secreted there. Your microRNAs and sort of exosomes and things are there as well. So all your cells are sloughing things off all the time. Whether you're in rest or in disease states, you change the expression of these things. And your proteins are there. So everything about you except for your DNA and your mitochondria is in your plasma, right? There's no cells and there's no mitochondria. But the environment that your genetics creates based on how it's functionally adapting to wherever you're exposed to gets captured in your plasma. So the idea was I can buy a cell that represents every organ of the body, right? We can buy quality controlled cells that are neuron, eye, immune system, heart, lung. So we basically created a human on a plate buying these cells from this commercial source. And then we populated those. We adopted the plasma environment from these different time points onto those cells. And we looked at the mitochondrial behavior of those cells. And we were able to see changes in unique cell types that then predicted at a specific time in flight when each of these cells would dysfunction, essentially. And so we were able to predict skeletal muscle dysfunction based on this, endothelial dysfunction, immune dysfunction, and we were able to get pretty close to the time where physiologically these things were happening in space. And so this then gives us a conduit to now scale. All you need is a blood draw, right? You don't need to do a muscle biopsy, which you can go to Quest or LabCorp or someplace else to do this, but there's still a barrier. You'd have to move away from your house. You have to drive somewhere. You'd have to interact with another human being to get this stuff collected.
God forbid. Yeah.
So where we've gone into this commercial source, and we've partnered with Versailles to do this in terms of the technology kinds of things, is how could we move away from that to a commercial space where you're in your home and you can get a self-collection done. And so we're down to these small blood cards, right? And it's got a couple windows in it, and it's 4 to 6 drops of blood in this first window. It wicks the serum out into the second window. The serum effectively freezes, right? So it's dried onto there. We've shown stability out to 2 months at room temperature. And so we've got huge scalability. Right?
Yeah.
And that's as far as we've tested. I'm sure we can go 5-6 months and it's still going to be fine. We then get this card in our facility. We extract the serum in our buffers, and you're off to the races with a NASA type test. You essentially have the metabolites, the exosomes, the proteins. You take that environment that someone's been creating for their time. You transfer it onto our muscle cell and other cell types that we can test in the lab. And we reflect then what's happening in you by looking at what's happening to this muscle cell. And you can go crazy, right? You can look at stress responses in mitochondria. And so the first part of the test looks at how your environment adopts onto this and creates a change in the baseline respiration. Typically, this is hard to convince biohackers and others, is being normal on our test is actually quite good. You don't want to be an outlier for certain measures. And so I want to be the best. I want to have the highest number. I'm like, dude, I don't think you want the highest number on the test for certain things. And so baseline, you want to be right in the middle where the cell is, and you don't want to perturb it in a negative or a positive way, which suggests that you're evenly balanced, right? And so we've seen this interesting thing. Biohackers tend to overstimulate in a basal setting. So they overactivate their mitochondria. So they're overhacking, right? And this is why you may not be getting the same return you expect.
I love that term, overhacking.
Overhacking, right? Be a new one. And then there's certain individuals that are dealing with crazy things like Lyme disease, long COVID, and we see an actual suppression of their baseline respiration, suggesting that they're creating an environment that's really not supportive of baseline mitochondrial functionality. And then we start messing with the mitochondria. You can add drugs in there to look at mitochondrial efficiency. You can add drugs in there to look at the potential of your mitochondria to create energy when they're stressed and tasked to drive as hard as they can. The second phase of the test looks at basic energy profiles. And so we're seeing whether your system is making energy using glycolysis, or whether it's doing it with mitochondria, and whether it's doing this in an efficient or an inefficient kind of way. So you get a stress response, you get an actual functionality, and then the third phase of the test looks at the dysfunctional element. The first part of that final test is a reactive oxygen species. So this is the other contract we've made. If you look at the evolution of organisms moving from water to land, the first pressure they had to adapt to in land was all of the oxygen that they were now exposed to instead of dissolved. It was an astronomically higher level. So there's a couple good books written on this, and there's some interesting papers on the evolution of biomembranes. It turns out that cholesterol appears in our membranes as we're moving from water to land. And cholesterol seems to be this adaptation to this high oxygen content. It actually binds oxygen and it tempers how it's delivered into our cells and our space. And so it becomes the biological calculus of preventing fire, right? And so to make fire, you need oxygen. To make molecular fire, which is reactive oxygen species, you need oxygen as well. And mitochondria are very good at making free radicals if they're not working well. And so typically there's multiple complexes in the mitochondria that move electrons. And one particular complex, if it's not working right, tends to add that electron off to something else, basically pulls it off of oxygen. You get a superoxide radical. The superoxide can't move very far before it reacts with something. It'll make hydroxyl. If it's in the mitochondrial space, you can get nitric oxide as well, and you make peroxynitrite. There's all these free radical species that then become toxic, and they'll start attacking your DNA, your proteins, your RNA, your lipids, and other things. And so we've developed a fluorescence-based sensor that can actually look to see in this same system, when we adopt your environment onto the cell, is that cell creating reactive oxygen species? And then when we stress the cell, is it suppressing them, or are you actually seeing a heightened level of these reactive oxygen species? So now you get one of the primary measures of mitochondrial dysfunction out of this test. And then the final component looks at the connectedness. So we as humans are more resilient when we're in a connected state, right? There's lots of research that's been done that people live longer if they're married, have kids, and have these, multi-generational relationships. The blue zones are a key example of this. If you want to live to 100, you don't necessarily have to move to Loma Linda or Japan or some of the other places. Just create an environment around you where you're surrounded by loving family and you do certain things. Mitochondria have a symbiosis as well. When you look at them in a cell, and one of the most amazing pictures to look at is any cell type under an electron microscope, where you're looking at it at, 5, 600, 10, 15,000 X magnification. You can actually see the structure of the mitochondria and of the membranes. And what's unique in every cell, that's like the heart and the brain and all these things, is the mitochondria are connected to each other, right? They form this symbiotic relationship where they communicate with each other, they transfer information, they sense their external environment. And every chronic disease we've looked at, you see this isolation of mitochondria. And so they separate from that network, and they're unable to sense and stress and adapt and interact, and they create dysfunction. And so we've developed a cell line that all the mitochondria in that cell glow green. And so then we're looking at it under our live cell microscope. When we adopt your environment, we then see if that network is maintained or if it starts to fall apart. And so we give you a network dynamic score as well. So by looking at all of these components, you're looking at a structural, functional, and a dynamic state of your mitochondria. So you really get a very clean, clear picture of every aspect of mitochondrial biology to then decide, do you need to go and do something to your mitochondria? Do you need to overhack them? Do you need to underhack them, right? It gives you a starting point to then start intervening on your mitochondrial health.
Wow. Okay. So now for the reactive oxygen species, before people flip out if they're new to the space, we do produce antioxidant, like the mitochondria also produces antioxidants. Does it? They do,
right? And
superoxide dismutase, right, which I know that some people love to talk about. Yeah.
Catalase. I mean, there's lots of antioxidants, as glutathione, all these things that are huge. The other thing is that it's not always bad, right? So reactive oxygen species get a bad sort of nomer in this. So if you have a lot of them, it creates injury. If you have a small burst, which is mitochondria are great at this, you can actually signal with this. And so this is where mitochondria become really complicated. They're these gatekeepers of a lot of processes. So when they generate a small rapid burst, it actually activates survival kinases and these other things in a cell that may actually be helpful to you rather than harmful. So not all reactive oxygen species are bad.
Yeah, well, I think we need some, just not too much. No, just not.
I mean,
we live in a what's the word? Who is it? We live in a Goldilocks world. Like our cells want to be living in a Goldilocks world.
Right. And a key example of this is nitric oxide, right? The Nobel Prize was awarded to the discovery of this free radical. And it does some amazing signaling things. And back in the day, I was in grad school when they awarded this prize. And everyone had these shirts on at these science conferences. What part of NO don't you understand? And it was, it's a crazy story of how that was discovered and how they figured this out. But bacteria use NO as a way to, or our cells, our immune cells actually use NO as a way to kill bacteria. Because they create this huge storm of nitric oxide, which then acts as a free radical toxin to these cells, right?
Yeah. So, no, all about Goldilocks. It's about, harnessing the good for good and the bad for good sometimes. Yeah. So, okay. Well, all right. So the Mescreen test is very cool. I've done 2 of them now, and I'll be doing a 3rd one in the next little while. But these cards are fantastic because you guys, if you end up doing this test, the first time I was exposed to a card like this, what it does is this paper absorbs it, and you get different components of the plasma separated out, right? They get drawn out.
Yeah.
And so you get a graduation, you have your red blood cells, then you get different fluids that dry out over the course of this this line, which is super cool. And it's amazing to me you can get so much information. So now, right now, your test, you had said to me, and I'm pretty sure I understood this correctly, but maybe I didn't. You said right now the way the test is maybe calibrated is it's measuring skeletal muscle mitochondrial function,
right?
Which is not the same as cardiac muscle or brain, or which now we're not talking muscle, but even cardiac muscle is different than skeletal muscle, right?
It is. And we think that skeletal muscle is a good starting point because you have a lot of it, and it's one of the major primary sinks for glucose and other things in your body. And so it's a good calibrator, right? And it also correlates to the muscle biopsies that we do because we're getting punches of skeletal muscle. We've shown good correlation between a muscle biopsy and the blood cards, at least in animal studies we've done. We're in the stages of planning a human study around this as well to compare what does someone's mitochondrial function look like in their skeletal muscle versus the same person that gets a blood card at that same time. Yeah. Which would be really cool to see. For sure. Well, I mean, that'll be,
I would imagine you've already validated the test, but at some level it gives you because muscle biopsies have been done for so long. Yep. Now you're putting it up against a known benchmark.
Yeah. I mean, we see a huge, in animal studies where we've gotten biopsies from muscle and then compared it to blood from that same animal. Yeah. We see an R value of 0.92, which has a huge P value of like 0.02 in terms of correlation between the blood and the muscle, right? So it's essentially the same thing.
Yeah. Okay. And so let's talk about the value to the individual. Yeah. Let's say somebody says, okay, I'm doing this test. I'm going to find out what's happening with my mitochondria. God forbid they get an over-hacking or under-hacking, whatever it is, result. Yeah. What can people do about it? Do we know? Because, there's so many things out there that I'm not going to name names, but let's say there's a million different things out there that say, optimizes your mitochondria. Yeah. Oh, this optimizes. No, that. No, this. And if you do this and this, oh my God, that's just going to set your mitochondria on fire, which, by the way, now we know we don't want. But nevertheless, what do we have a list of things that people can do that is helpful to them? Or
there are lots of papers that show different elements of mitochondria and how you could perturb ROS, how you can change mitophagy, how you can do these other things, right? And our test predicts some elements of that. And so the problem with this is we can predict and give people suggestions, and our algorithm will do that, right? It'll give you some ideas of where to start with your hacking space. One of the things that we can't predict is how your individual body is going to respond to this. This is a problem we're working on right now, is how do we then test things that will respond to your physiology, your genetics, your biology. So like you said, there's 1,000 million ways to hack your mitochondria, but I think it's unique for each individual. Mm-hmm. Where we see this test, it gives you now the power to determine whether something is working or not and how you tweak that. And so I get asked this a lot of times, who should take this test? And anyone who's living and breathing should take this test because you should know what your mitochondria are doing. And if they're cooking along just great, don't change anything. Do that. Test them once a month or once a year and you'll know. Yeah. If they're not working in an optimal way, then you know where to start intervening. And so I've talked about different places of what to do. There's really 3 things to do to your mitochondria, but these 3 things can be done in a million different ways. The first is, and what most people need, is lipid support, right? They just don't have a lipid architecture that supports the behavior and necessity of what mitochondria need to do. Mitochondria have a multi-layer lipid structure. They have an outer membrane, an inner membrane, and the space between them is critical. And so all of this is lipid regulated. And the other aspect that people don't really think about is most of cholesterol synthesis happens in your mitochondria. And so people who are worried about hormonal changes, and I know men are taking testosterone, all these things, just make your mitochondria better, right? All of that cholesterol is the core structure for all of these hormones. Yeah. And we think that this will stabilize a lot of these things. And so mitochondria also have their own lipids. Cardiolipin is a huge thing that people talk about, and oxidization and all these other things. And so you need to make the structures of your lipids better, not only your plasma membrane, but your nuclear membrane, your mitochondria. You can supplement this with phosphatidylcholine, eat, foods rich in good cholesterol and those sorts of things. If you're going to eat the egg, eat the whole thing, not just the whites, the cholesterol in the yellow part.
But also, what do you think about eating I was taught, and I'm a believer that egg yolks are it's a better functional food if they're runny than if they're hard boiled.
Yeah, possibly, right? Then you're not denaturing all the lipids. And things.
You're not messing with the lipids as much, kind of thing.
You just go to Japan and have raw eggs, right? I mean, this is
I can stay home and have raw eggs. I used to, I'll occasionally spin a raw egg yolk, not so much white. Yeah. But I'll spin the egg yolk into, a coffee or into a shake. And it just gives you that.
Foods that definitely have this, right? Shrimp have really high cholesterol content as well. And so there's ways to find foods that do this and supplements that do this as well. So once you've built a better membrane system, I think a lot of problems will go away, right? And so this is the base that we tell people to start at. If their mitochondrial test reveals that their lipids seem okay, then we move to the next phase, which is really getting rid of death, dying, and decay. So this is where you want to activate autophagy and mitophagy pathways. There's lots of ways to do this. Intermittent fasting is one of the easy ways to hack this with very little cost. Mm-hmm. Hypoxic breathing will do this as well. And so there's lots of endogenous systems that get activated. And then there's compounds, right? We've discussed urolithin A. There's, people who are more extreme will do rapamycin and things like that to activate the autophagy, mitophagy kind of networks. And this really gets rid of the things that aren't functioning in the proper way and recycles them and re-energizes the raw materials back into the system. And then the final component, I really think then once you've stabilized your membrane, done this mitophagy, autophagy activation, is to balance the antioxidant system, right? This calculus of always having your mitochondria churning and working is as they work more and more and more, they're more prone to make free radicals. And so you have to balance that pool. And there's ways to do that as well. I mean, this hypoxic breathing, I think, is magic. Can you
describe what hypoxic breathing is for the audience, just so they?
Yeah, so we've been.
What is he talking about?
So we're doing a study right now on campus where we're doing teaching pre-med students who are quite stressed. We want them to sleep better and do other things. And so one of the things we're trying to teach them in this protocol is how to do hold their breath. And the idea is to do an extended hold. So within a week, you can teach someone to hold their breath for about 1.5 minutes at a time.
Really?
A couple times a day, right? And it's with meditation and other things. So it really activates this thing. So my PhD thesis was looking at how we could protect the heart from a heart attack. And so in the mid-'80s, it was discovered they were actually trying to create a huge injury in this large animal model. And so the thought was, if we create some small bouts of injury before the long heart attack, we're going to get this massive heart attack that happens. Mm-hmm. Well, it turns out those small bouts of injury created a preconditioning-like effect, and the heart was magically protected from that huge heart attack. Turns out every organ has this endogenous system of preconditioning, and it's an acute program of protein movement and phosphorylation. And it's a long-term effect of new protein synthesis that regulates this process. And so my PhD thesis was to look at the second window, the longer-term phase of this, of how these unique stressors create this protective phenotype. And so I think philosophers always get it right before the physiologists get to this, right? And so Nietzsche had said, that which doesn't kill you makes you stronger, right? Yes. And so, and musicians have said this all as well. And so this idea, let's possibly, it's sold from Nietzsche, I think. But if you create this toxic thing in an organ, but you let that organ then adapt to this toxic thing, it remembers, right? There's a memory phase of this, and it lasts almost 72 hours, and it launches an entire program of resiliency. And so we think this is what hypoxia does. When you make your cells and brain and organs deprived of oxygen, you're activating this endogenous system that exists in every one of our organs. And it now learns to adapt for an extended period of time to that stress. And so then if you were to have a heart attack, or if you were to have an injury, or you get this remodeling effect that happens at a much faster rate. Turns out a few years later, these guys figured out that what it does is it actually stabilizes the membrane, which then in turn stabilizes the mitochondria. Right?
Back to the membrane,
and it's energetic as well. Your energy is completely preserved during this phenotype then.
Well, we're talking about hermetic stressors here, really.
Yeah.
Right? Like we're talking about a stressor that's strong enough to elicit change and stress, but not strong enough to overcome body systems, but it pushes the systems to adapt. I want to go back to something you've mentioned a couple of times, and that's cholesterol.
Yeah.
Because, you know, the current world that we live in the allopathic world, it appears that there's this driving race to the bottom when it comes to cholesterol. Right. Oh, your cholesterol's too high. Well, wait, right? I mean, the numbers keep dropping and dropping and dropping. And I know that I've seen at least one paper, if not more, that talks about in 80 and 90-year-old people, the lower their cholesterol numbers are, the higher the rate of depression and dementia that we see. Like they can't function. And yet somehow we've got a system that continues to harp on this cholesterol is bad story. Right. Can we just go back to that cholesterol for the memory?
I think cholesterol free floating in your blood is not a good thing, but when you start lowering that, the potential to lower the cholesterol that's good in your plasma membrane goes up, because those drugs will start pulling that cholesterol out as well. Yeah. Especially like the really sort of potent statins and things like that. And we've done studies in animals. We've started to look at humans in this space as well, where statin treatment may not be optimal, right? And other things because of this particular effect on membrane-derived cholesterol. So for the last 20 some years, my lab and our collaborators have focused on a specific part of the membrane called the lipid raft in caveoli. And so they create this unique structure in the membrane that's different from all other parts of the membrane. And the unique biochemical feature is it's highly enriched in cholesterol, sphingomyelins, and proteins. And so we think that signaling and efficiency that we create is really platform driven, right? It's not this fluid mosaic model of the membrane that was initially thought, where things have to randomly collide and come together for signaling to happen. Turns out that biology actually creates a platform, and this creates the efficiency. So now in this really tight space, when you pack lipids in, this is where everything likes to sit, right? Things like fat at the end of the day. And now you have your signaling molecules, your receptors, all of the effector things. Turns out we had published a paper where your mitochondria sit right under this space as well. And so now you have this conduit for all the signaling, but the energy regulation happens in this tight conduit as well. Wow. And so it's cholesterol driven. So once you start depleting cholesterol, this membrane starts to become more fluid, less tied to these regions. All of the stuff that's there starts to float apart and gets dysfunction. So we've got a couple patents and things around this. So we actually have a marker protein that drives cholesterol recruitment to the membrane and creates more of these lipid structures. And your idea about depression, anxiety, all these things, it turns out that it may be a key regulator of neurodegeneration. We have a series of papers with my collaborator, Brian Head, that we've published where Brian has shown that it's critical to Alzheimer's phenotypes as well as ALS. And there's a company called Iconoclastes that actually licensed this patent, and they're developing this as a first-in-man gene therapy for ALS. And we're hoping to be in humans in under a year. And it restabilizes the membrane architecture, right? It makes more of these cholesterol, lipid raft, caveolar domains in neurons. And it goes back to this first principle of mitochondrial behavior. Put the lipids in and everything will come back, right? So it turns out that if you have a neurodegenerated state where the membrane and these structures are falling apart, you add this one protein, the structures rebuild, everything comes back signaling-wise, right? And all it takes is this one protein that's driving this cholesterol movement into the space. And it comes down to cholesterol, I think, is critical to life. We've jokingly been saying in the lab, right? If you're having a heart attack, you should go to McDonald's and have the fattiest meal you possibly can.
No, that's not good fat, though. I mean, I think we have
you
to
want a cholesterol load when you're going through this response.
Okay, but maybe not McDonald's.
Maybe not that, right?
Maybe pound like a few raw egg yolks.
Or a shot of olive oil or something.
Or a shot of olive oil. Exactly. Yeah, because I was going to ask you, like, what your thoughts are on this? You know, there's a huge discussion around damaged fats from foods that are fried, fats that are overheated, you know, the wrong, like unhealthy PUFAs.
Yeah.
The seed oil thing is turning into like an inferno because some people are screaming, no, seed oils are going to kill you. The other side's like, what are you talking about? And maybe the truth is that seed oils that haven't been properly managed are the problem.
Yeah.
Like, you know, again.
Well, I mean, and I think nature has a lot to tell us about oils and things, right? So one of the things, we have normal olive oil that we have in the kitchen and stuff like that. But if I'm going to put olive oil on a salad or something that isn't cooked, I tend to buy it from Morocco, right? Mm-hmm. There's this idea that those plants have invested a lot of energy into those olives because they live in a really harsh environment. Yeah. And there have been studies done that show that the polyphenol content of those things is astronomical compared to stuff you can buy from Italy and other places, and even California, right? They just don't have that stressful environment. And I feel way better when I eat that oil on a salad kind of thing rather than do stuff with it. So
what about Greece? Because Greece is pretty arid. I think Greece is
it is, and I can't think if I've seen the paper that compares them, but I think there have been studies that have compared different olive oils and their polyphenol content, which I think is a critical thing to look at.
Okay, well that's really interesting. I'm from Morocco. I should call somebody.
Yeah. I think the one I recently got was Atlas, which has a couple other versions of it. But I've heard it's amazing, and I feel amazing after I eat it. So,
all right. Well, good to know. Okay, so I wanted to come back to the cholesterol. So much good stuff here. The lipids, hypoxic breathing. Hypoxic breathing, just something so simple, right?
Yeah. I mean, you could literally sit in your office for 10 minutes and create a preconditioning-like effect throughout your body.
Yeah, just if you're getting lightheaded, take a breath. You'll get better over time. Don't pass out.
And most people hold their breath on an inhale. You actually want to hold on an exhale.
Right, because although on the inhale you've loaded up with oxygen, right? Yeah. On the exhale you've already depleted the oxygen.
And that's where the people in a mindset, right? They're like, I gotta get as much in as I can, and then I'll hold my breath, and it doesn't work as well.
Well, I think if you watch free divers, don't they exhale before they go?
Yeah.
Right? They're creating. So those guys are the hypoxic kings and queens of the planet. It's
amazing. Yeah.
It is amazing what they can do.
And they get that pressure change as well when they're diving, right? So there's lots of physiology things going on there.
Okay. So now let's talk, let's go to the cool stuff.
Yeah.
Let's talk about the things that, I mean, we already touched on cool stuff. Like the hypoxic breathing is, guys, it's free. Like, stop eating so much, so often. Like eat less often, breathe sometimes on occasion less.
Yeah.
These are two very powerful free strategies. Get very hot, get very cold. We didn't really talk about this, but these are both stressors. Done right under the right circumstances, these are hormetic stressors that will improve everything and stimulate mitophagy and autophagy and all those things. But there's something else that you've been studying that I think is really interesting. And it's not, I think to a lot of the audience, they've heard, some of them are going to be very familiar with it. Others are going to be like, come on, again with the meditation thing, just stop it. I can't meditate. And you have a really interesting way to meditate. So maybe share a little bit of the work that you've been doing around Dr. Joe Dispenza's retreats.
Yeah.
So
I shared an office with Toby Muller-Bertram, who's a pain anesthesiologist. And so the weird social experiment we were doing at that time was put a PhD in the same office as an MD. And so at the end of the day, they'll start talking and they'll rub off on each other, right? So I'll learn a little bit about human stuff and he'd learn about some research things and we would solve the problems of the world.
Right.
We sat in this office for almost 10 years together and we became really good friends, but we never worked on a problem together.
Come on, for 10 years?
For 10 years, for a decade at the VA hospital on campus. And so then Toby left, started his own pain service in the Palm Desert area, very successful. I think it's like an insane number of clinics, like 15, 20 clinics now. When the opioid epidemic hit, they started experimenting with alternative ways to manage pain in the clinic. And one of the practitioners, Hillary Hamilton, who was there, was a Joe Dispenza person, and she was playing around with his meditations with their patients. And they saw some interesting pain effects on these individuals, less pain after they learned how to do this process. And so Joe had visited his clinic. Then sort of Toby forgot about this, like most of us do, right? Who wants to meditate? Who wants to go learn it? So about 4 years later, he ends up at a Joe event. And and Joe describes this as, how do you describe the taste of a mango to someone who's never had a mango? And it's like, you're just eat the frickin' mango, right? And so this is, you can't describe a Joe Dispenza event in words. I've tried, and people, roll their eyes, or like, oh, whatever. You just have to experience it, because there's so many things that happen at an event that will take you on a journey of discovery and all these other things. And so Toby discovered a lot of things at this event, and he went up to Joe and he's like, we need to study this, right? I mean, it's this perfect petri dish where people are away from their lives. They're on the same sleep-wake cycle. They're eating essentially the same food, because it's, you get a breakfast, a lunch, snacks. The only thing you're on your own for is dinner, but you're at a resort, so there aren't many options. Mm-hmm. And they're captive. I've never been to an event where you pay to go to this event, so you're there, right? So people wake up, they're listening to the lectures, and I've given a lot of lectures, and people are always on their phones and doing all kinds of crazy stuff. At this event, they're intent, and they're writing stuff down, because they're absorbing, and they're students, right? They're absorbing this information. And transformative things happen. I mean, Joe has, if you go online and you look at these stories of transformation, they're essentially these case studies, one-off kinds of case studies in every disease you could imagine, where someone has transformed in a unique way by doing this work, either on site or finding his books, reading his books, and doing his work. So Toby sort of went up to him and said, we need to quantify this in some way. And he's like, I know a guy. And so I'm the guy. So he was listening, right? For those 10 years, he knew all the stuff that I could do. And he's like, we need that in this space.
Yeah.
And so we got together. There's a donor that had come in, and she really wanted to move this to the next level. And someone, without consulting me, had thrown out a number. I'm like, I would have given it a couple more zeros, because science is expensive. But it started us on this path, and we discovered some stuff. And we did a study. First study was Indian wells in 2020, and this was in February of 2020, so right before the pandemic. And then the samples came to my lab, and we were essentially shut down for a couple months, and we sort of lost track. And I'd get these calls from Joe. He's like, hey, get a chance. I'm like, who the hell is this Joe Dispenza guy that keeps calling me? And we finally looked at the data, and it was astonishing, right? We saw changes in 7 days in individuals that we would have never predicted. Wow. And so he had been trying to get me to come to an event and eat the mango for about 12 months. And then I saw this data. I'm like, all right, I got to go. Yeah. And so I brought the postdoc in my lab that was doing this work as well. We were doing a study in Orlando, but we immersed ourselves in the retreat. And I have never cried this much in my life. I mean, it was a cathartic, crazy, amazing experience. My mom died suddenly, and it was this trauma that I've always had, and something that was a core part she was a huge part of my life. Mm-hmm. And to lose that without having this ability to say goodbye and all these things, there's a lot of trauma associated with that. And this kept coming up for that whole week, and sort of resolving and seeing this, right? And so then we designed some studies that have just gone far out. There's a documentary that came out called Source. You can go to, I think, sourcethefilm.com and see it. It's through this nonprofit that sort of developed this. This gets you the early look at what we were doing. Since the data in this documentary, we were light years ahead. We're doing studies on thousands of people at a single event, and we're looking at how there's a community connectedness that forms, and this idea of an environmental pressure that's created, that Joe creates in this week-long event, mm-hmm. and how this then converges with our genetics and our mitochondria and our biology to cause this transformative change in individuals. And so we're looking at epigenetics, we're looking at transcriptomics, we're looking at microbiome changes. And I have yet to be disappointed, which I'm shocked at, right? Eventually something's going to go wrong. But everything we look at, we see something that's just phenomenal. You'd never predict it. And in 7 days, you see this explosion of metabolites in people who immerse themselves in this compared to controls that are hanging out at the pool drinking Mai Tais all day.
Wow.
It's not relaxation. There's something happening at this event that you go through this transformative change. And it is, the way I've been thinking about it is that there's toxic ways to do psychiatry and psychology, right? If you have negative thoughts, negative emotions, if you're exposed to traumas, you create these multiple split personalities that allow you to deal with this. And so what I think is happening at a Joe event is he's actually creating a positive personality. You go through this and the room transforms. So you see I was talking to someone yesterday, one of my favorite things to do at a Joe event is to get there early and just sit in the front lobby and watch people as they're checking in at the beginning of the week. And they're assholes, right? I mean, they're upset with the world. They're screaming because they've had, flight delays and these other things. And it's just comical to watch this because in 3 days, that person's going to be giving everyone a hug in the hotel. And it's just this environmental shift you see. And it's because you're introverting and you're creating this social dynamic environment around you because you're absorbed by this crowd of 2,000 people that are creating the same energy. And the entire human body shifts. And we're seeing this over and over and over again. And when you look at the 1,000 people studies, it's not that one person is responding. We're seeing almost an 80% response, and they're shifting in almost similar kinds of ways. And so we do a lot of surveys at our events. And so the surveys are very revealing. The SF36 asks you 36 questions about physical, mental, emotional well-being. The biggest shift we see, irrespective of disease, is a mitigation of pain, right? And so when people's perception of pain changes, their body and their dynamic and their feelings and their outlook completely shifts. And we see this across the entire population, which is just crazy.
Wow. I mean, I think we so underestimate the power of the mind and the power of intention and the power of breath and the power of energy. And, there's nothing like a doubting Thomas like yourself, who is so well educated, who's so steeped in your craft, to once he's had the mango go, oh my God, that is the thing.
Yeah.
Oh my God.
And I mean, this is, we're always looking for an external thing, right? And we're willing to pay thousands of dollars. Why not train your mind and your breath and your things that are free to do all this, right? We have the tools and we've been invested with those tools. We've just forgotten how to use them. And I think what Joe and these other individuals teach you how to do is to re-engage with the tools that we're naturally born with and to use them in a unique kind of way.
Yeah. No, I love that. And it's funny. I don't think that it makes everything else redundant necessarily.
True.
But I do think that not tapping into this piece of
I totally agree.
Can thwart the best other stuff.
Right.
And prevent, right? I mean, if you look at some of the case studies that Joe Dispenza has, it's mind-blowing, like healing from cancer or, and it's not like, oh, everybody does this meditation for 30 days and your cancer is going to be gone. Right. Not at all. There are individuals who have tapped into this and made it work for them.
Yeah. And I totally agree, right? I don't think you can completely absolve yourself of Western medicine and other things. I mean, there's this joke in religious circles where there's this guy and there's a flood coming and he's, there's a jet ski that comes by and is going to take him away. He's like, don't worry, God's got me. Yeah. And then there's like a boat that comes and he's like, don't worry about it. And then there's a helicopter and he's like, I'm going to be fine. And then he drowns and dies and is at the pearly gates with Peter. And he's like, I did all the things I was supposed to as an individual. I believed, had faith, I did all these things, yet God let me die. And Peter's like, God sent you a jet ski, a boat.
That was God, by the way.
And this idea, right? And so there are tools that I think faith, divine, whatever has allowed us to create as humans. And all these things that Western medicine uses, I think, are the synergistic things that could be augmenting the meditation, the breathing, and other things. And so as a pharmacologist, I would never tell you to move away from pharmacology. Yeah. But I think having a happy medium between the two and accentuating all of that, you're going to get a huge synergistic effect, I think.
Yeah, no, I love it. I couldn't agree with you more. I mean, I think that the next thing, the last little thing I want to unpack with you is there's so much resistance to meditation, and particularly with Dr. Joe's work.
Yeah.
His baseline, I think, is like an hour in the morning and maybe an hour in the evening. Like it's a massive investment in time. And you could argue there's no better investment you could make, but people's realities are what they are.
Yeah.
You have a really interesting story about how you meditate, which is it's not a traditional meditation. And what I'd like to maybe offer people is that there's different ways to get to this meditative space, but you have your own way, which I think really illustrates the fact that maybe you don't need to sit there for 2 hours a day, unless maybe you've got something really big going on.
True. And I learned this by going through a week-long experience, right? And so this is where you do have to invest some time into self-care. And I think attending a retreat and getting this experience of seeing what works for you is a good way to do it. And listening to the lectures, right? I would just sort of drown out everything and just take some information in. And one of the things that's critical to Joe's sort of process is this concept of self-reflection, this idea of mental rehearsal. He teaches you how to do this in a unique kind of way where you basically are meditating with your eyes open, with a lucid kind of dreamlike state. You're never asleep. You're completely aware, but your mind is in this outer space kind of thing, and you're experiencing mystical things and other stuff, right? Athletes do this, right? They have this hyper focus, this ability to basically turn everything else off and to go into this space where they're rehearsing every activity they're going to do when they're in their sport. And this is why they're at this elite level, is because they have this capacity.
Yeah.
And so I do Joe meditations once in a while at home, and like you said, the hour ones are tough. I'll do them. We have a Thursday group that meets every Thursday, and I love doing the meditations with that group. Joe has these series of 15-minute generating kinds of things. And if you practice, those will give you the same sort of achievable kind of thing. But again, you have to find concerted time to do that. So how could you build a meditation into your daily routine kind of thing? And so we had these new devices that we bought. These were these patch devices where you put 3 electrodes on your forehead. We're going to use it to track sleep, but it turns out you can actually track meditations and things with this as well, because you get some frontal lobe kind of measurements of electrical activity. So I tested one out at home, and I collected my data for the evening, dumped it on my computer, sent it to our collaborator here who's doing the analysis. And Julie called me and she's like, "Do you meditate at night when you go to bed?" I'm like, "No, what are you talking about? I just sit there and do stuff and then I go to bed." She's like, "It looks like you're in this weird gamma state for like almost 45 minutes to an hour. And then you go into this deep sleep and then you do the normal thing throughout the night." And I'm like, and so I tried it again, and the second night, same thing. And so one of the things I do, and I do this instinctively, and I think I've done it when I was younger, and I think I do it more after coming through a Joe event, is I will mentally rehearse at nighttime, right? I'll think about all of the things that have happened in the day, how they're impacting my life. And one of the problems that I've learned in the Joe world, and my wife teaches me this all the time, she's into mindfulness stuff as well, is that we're constantly stuck back there or in the future, right? We're not right here. And this is the problem. And so I use this time to get rid of the back end, the memories of things. And then I use this as a time to focus on things that are coming, and I start mentally rehearsing for those. But then I'm completely in this present moment thinking about those two things and how they sort of equate. And my mind naturally goes into this gamma-like state where I'm hyper creative. I have all these ideas and thoughts and things. And then my mind exhausts and I just fall asleep, right? And it's so every night that you do this. And there are nights where I don't, and I feel more tired in the morning because I haven't had that ability to do it, right? You notice it.
Yeah, it's almost like you're processing and releasing and cleansing your mind and getting ready for the next day. So what's your mitochondrial function like?
It's pretty normal. I mean, I've seen
that's
it's
right.
average,
What you
right?
want, right?
It's average.
You be in the middle. Okay.
Yeah, I'm in the middle. I have a potential that I can drive. So there's certain numbers you want to get higher, right? And my ROS levels are in the optimal range when I'm baseline and stressed. My networks look okay. My potential goes up, but there are things, but you have to keep working at this, right? I think your mitochondria are very dynamic structures, and their functionality is very dynamic as well, in the standpoint of you can mess them up in a few months and you can make them better in a few days, a week, right? We've done some studies with mitochondria in the Joe world, and you can completely change your mitochondria in 7 days if you go to an event, right? They look very different from where you start to where you end up.
This is not an ad for Joe Dispenza's event.
Right, it's not an ad for his event. But this is where I think one of the big barriers for people getting into meditation is they think that it's going to take them 30 years to become this practiced, disciplined, Zen Buddhist meditator. One of the things we've seen with the metabolite analysis we've done is that a new person going through this immersive retreat really becomes an experience like meditator in 7 days. And we've shown this to psychiatrists on campus, and they can't believe this blood transformation that you see. And you can't fake this, right? Because you're not, I mean, it's a biochemical environment that you're creating. And so I think there's deep ways to do this. I think the problems with most other ways is, and I think there are some guided approaches. I think you really need to be guided and need to be taught. And this is what I think is masterfully done, is you're taught a certain thing and then you're putting that to practice. And we do this in our worlds as well. And so I think there's a real sort of concerted element to that week-long event. But there's other ways to achieve that as well. We've seen people that just read as books and they have these transformative experiences, and they're just gaining that knowledge, and then it's work, right? You have to immerse yourself into doing the work. You can't just expect it to fall out of the sky, I think.
Right. And so have you seen anything else produce such results?
We're starting to look, right? We're starting to do some comparative studies. In the literature, there's lots of papers on Vipassana and those sorts of things, which is very breath focused and Kundalini and all these other things. And I think there's elements of this. We're starting to do some comparative kinds of things. I think mindfulness has acute effects, less chronic effects kinds of things. And so I think there's probably a dosing thing as well in all of this and one of the critical things we're starting to look at is once you leave the event, how long does the effect last? How often do you have to do the practice of all of this stuff? Can you essentially quit cold turkey and you're good for 2 months, 3 months?
Right.
What if you did this once a week? Can you sort of keep it up? And
what's the minimum effective dose?
Yeah, we're starting to look at the effective dose kinds of things.
Okay. Well, no, and what I was asking is, is there anything else like fasting or is
not at this level?
It doesn't sound like there's
so many things that happen when you close your eyes that we don't think about.
Yeah.
One of the other things that I sort of, after going to my first event, one of the things I realized is other than sleep, I don't close my eyes really. And sometimes all you need to do is turn the lights off in your office, close your eyes for 10 minutes, and you're in this completely transformed state after that,
Yeah.
So,
well, I have a program that I do. It's called NewCalm. And one of the things, and NewCalm it's a play on binaural beats a little bit.
Okay.
And, they've used music to wrap around it and then this binaural beat track. And they have different formulas, if you will, to get you into to get you into different states.
Yep.
But a big piece of it is an eye mask.
Yeah.
So it's taking the stimulus from the outside out of play. And then they use a biosignaling disc on the wrist, which is, another piece of the puzzle, if you will. But this whole idea of shutting out the outside world is a really interesting concept.
It is, yeah.
And crazy that we went from mitochondria to this and back to mitochondria again. Because at the end of the day,
it's all about your mitochondria at the end of the day, right?
This is what it's about, right? Yeah. I mean, it's so interesting. And so, you know what, like to the audience, like you could do your own experiment. You could get your mitochondria measured and then take up meditation for 30 days and see if it makes it, but really commit to it, right? I think it's not a, but like you said, I mean, maybe mindfulness or certain types of breath work. There may be many different ways to skin this cat. But how fascinating that we finally have a test to measure mitochondria, because it's been one of the most elusive things. Like you can measure almost anything else, just not that.
Yeah. No, it's been great. We've been seeing some amazing results in certain people that we've put on a path to fixing their mitochondria, and some of them we haven't, right? You just haven't gotten the equation right yet in them. And we're the next phase of what we're doing, I think, will help solve that. And the idea is really to get personalized mitochondrial health. And one of the things we're envisioning is, could you recreate that individual over and over again and start testing different compounds and things in our system to really move them down that path? Right. It's time consuming and expensive to do, but it's the next wave of personalized mitochondrial medicine, I think.
Which is medicine, personalized medicine. I mean, ultimately. And so at this stage of the game now, when people do their test, do they get any kind of coaching or any kind of feedback? Is there someone who's available to help them make sense of their tests and maybe make sense?
So our dashboard has a lot of that information in it, and the algorithm will suggest some things to try out and play around with it. You can always book a consult if you want your handheld and sort of people to walk you through that report, but we're making it simple enough that you could just read it, understand it, and get some suggestions to move to the next phase. And it's an education thing, right? So we're going to have an education platform that teaches people about the cell, the mitochondria, how it relates to the nucleus, and all these kinds of things. So they'll be able to edify themselves. And I think we need to make a better consumer. We need to teach them what they're looking at and why this is important. And then I think at the end of the day, one of the things is mitochondrial health. You really need to be followed by someone who is taking care of your overall health. And it's a key picture of everything else. And so we have a lot of providers we're working with right now as well, who we can funnel people into, and they do all kinds of holistic therapy, MD, Western medicine based therapy, all extremes of it, right? And whatever people feel comfortable with, I think, at the end of the day.
Love it. All right. Hemal, where can people find you and find out more about the test and get their hands on one if they want to, or where do people learn more? I could keep talking for as usual.
I can talk forever as well. Mescreen.com gets you to the info landing page. And then you can follow me on Hemal Patel, PhD, as my Instagram handle. Every Monday we try to put out a unique mitochondrial fact. And then, I share weird stuff about things that are happening in the lab.
Perfect.
And other crazy things that I do at home. Like my wife had just bought this new camera, and so we were screwing around and I took this amazing picture of the moon. And I got to share this, right?
Yeah.
So stuff like that. But really abreast of things that are happening in our space and in the world and the mitochondrial space, as well as things we're doing with Joe and really expanding into this other area of mind-body kinds of relationship. So that's the best way to keep track of all the things going on in my life. But mescreen.com will get you to the test and the platform. Yeah.
And I believe we have a code for people which is longevity, which will possibly help them, give them a little bit of a reward for being a listener of the podcast. And that's it. Hemal, this has been so fascinating. Thank you so much for taking the time.
Great. Yeah.
I personally will be following you constantly like a stalker. And I hope maybe to see you at a Joe event sometime, because I think that's on my list for 2025. This year was one guy. Next year, I'm moving on to Dr. Joe. Just keep building the inner resilience muscle.
Good. No, it's a crazy, amazing experience. And there's so many crazy, amazing things in this world to do,
100%. Thank you so much.
Yep. Great talk.